Okay, well, I'm really pleased that we have some contributions from scientists at other light sources. And now we're going to hear from Sam Webb, who's a lead scientist at the Stanford Synchrotron radiation light source. He runs the scientific programs at the XRF microprobe and microspectroscopy beamlines. He has a strong background in environmental chemistry and geochemistry and worked with a variety of different experiments across many fields from the geo, bio, and material sciences. So he's going to give us the first of our facility updates from the SSRL, the Stanford Synchrotron radiation light source. And Sam, I think you're all set to go.
Okay, can you see my slides there?
Clearly, yep.
Okay, excellent. So I'm going to give you a brief overview of the SSRL Earth Science Research Program, and hopefully just give you a flavor of the types of stuff that we are doing at SSRL that could be relevant for the types of stuff that you might be interested in. Let's see, here we go. So SSRL is a beautiful synchrotron out here in California. We currently have 17 different branch lines that cover 33 different experimental stations. So performing typically 33 different experiments at a single time. And you can see our breakdown of beamlines kind of into the bottom there, the structural molecular biology scattering in terms of crystallography and small angle scattering. Several material science beamlines for vacuum ultraviolet type spectroscopy and hard X-ray scattering. And then our chemical catalysis beamlines, which kind of cover spectroscopy and imaging. So many different flavors of different experiments. Pardon my dog there. Anyways. I'm going to talk to you mostly about our imaging beamlines, since that's my specialty at SSRL, and talk a little bit about spectroscopy as a whole. So you've heard a lot about different spectroscopies from Joanne's talk this morning. And a lot of what we often will do are, these types of time scale spectroscopy experiments that you saw in Ludmila's talk earlier, and the types of stuff that Michelle was talking about with the total scattering too. And here we have looking at ferrihydrite sulfidization and looking at iron sulfur nanoclusters that form. So adding sulfide into small ferrihydrite particles and watching the degree of sulfurization as a function of time, and be able to characterize the types of both sulfides that form, aqueous and solid phase type species. And this is one way to do a lot of reaction type experiments, but we also, again, as Mark was showing earlier, can do this in real time too. This is a set of experiments that we did with a group from Caltech, and actually looking at a biogeochemical reaction where we took manganese 4 oxides with bacteria in a flask outside the hutch, inoculating that with a Shionella and watching the bioreaction and watching the solid phase changes that occur through time as we go through the biochemical reaction, and seeing that as things go from manganese 4 to manganese 3 to manganese 2, depending on the solution and the chemistry of the solution, you can see rhodochrosite being formed, or seeing manganese 2 staying in an aqueous form, depending on the saturation conditions. And getting this in a complete time series. So taking a full Zane scan every 10 minutes or so and watching this progression happening. We also took out aliquots to do for X-ray diffraction as well. So in a lot of cases, we can combine these types of experiments with diffraction to get a total picture of both the near edge spectroscopy and the crystallinity and structure. I'm going to shift over to our imaging program. Our imaging program at SSRL is primarily accomplished through 4 different beamlines. And again, our focus here is really on spectroscopy as well as the imaging parts. So you kind of need to have those 2 pieces together that really give you a lot of information for your environmental and geochemical type studies. We cover a wide range of energies from 2 to 25 keV, and spot sizes from 1 to 500 microns, to go from the very small scale to the very large scale. So, and, you know, on the biological side, we can cover from the cellular level up to the organism level. You can see everything from cells up to whole hamsters here. In the same kind of sense for a geological system, we go from individual grains and work our way up to large cores. And I'll talk a little bit more about that in a minute. Also with new upgrades that have been going on recently, we've removed a lot of our microfocus KB optics and upgraded these to SIG-ray, axially symmetric optics. This has really given us a large jump in performance. We can now reach smaller micron and submicron spots, and we have a higher flux in those regions. We've also combined that with hexapod systems to be able to move between different spot sizes very quickly. So we can go between, say, a 5 micron and a 1 micron spot reliably, like you would change the head of a light microscope in the laboratory. These optics also have a very narrow depth of focus. So we also have a laser arranging system there to keep the sample at a constant focus. One of the neat things about this laser system too is it actually does output the topography of the sample effectively. And so in a multimodal sense, we actually record the height changes of the sample as well as the X-ray profiles. And it's actually kind of interesting to see when you have some interesting samples, and also lets you kind of gauge the quality of your research groups and see how well they can polish their samples down. So it's kind of a, I say, unofficial challenge to see who can make the flattest and most nicely polished samples. 14.3 is one of our premier low energy beamlines, tender energy beamlines. Has those optics in there. And this has been a really important one for doing spectral microscopy and combining this with other techniques too. So you can see in the bottom right corner, this is one of the first results that came out of the beamline with our new optics. And this is trying to correlate the sulfur distribution with a FISH, a fluorescent in situ hybridization map. And so you can see the different populations of bacterial cells in the green and the red, and correlating that with the chemistry, sulfur chemistry that we see in the other profile there. And this is just a kind of a first profile, but now we're doing more proper registrations to really get a good image and seeing what populations of bacteria are leading to what changes in chemical speciation of the sulfur. And we saw a little bit about phosphorus in the last talk. We've also been doing this with phosphorus speciation in soils too. This is with work collaborated with folks at the University of Delaware. And again, comparing both phosphorus in soils and comparing that to the iron speciation and trying to correlate changes in iron with changes in organic and inorganic phosphorus. Now, one of the real powers of having a spectroscopic imaging beamline is being able to map at several different energies through your XAS spectrum. So we can create these different images at several different energies. This is for sulfur here. And sulfur is a great element, my second favorite element behind manganese, because it has a wide range of species that are distributed here. So you can see the full oxidation states from -2 to +6. And there's a lot of chemistry that we can tease apart in that here. And by mapping it at the different resonances that are indicative of those species, we can actually put together a map, a chemical map. And so as much as I love staring at squiggly lines, it's really nice to be able to see this in a full resolution, in a full field kind of way. And as an example here, this is some work done in Colleen Hansel's lab, looking at sulfur speciation in corals. And what's really impressive here is being able to now look at these light microscope images here that show the structural calcium carbonate with the organic material in the coral here, and seeing that light up in a fluorescent microscope, and then I'll be able to compare that to the different sulfur speciations. So we can see the sulfate in the carbonate matrix and all different forms of organic sulfur as well. And really be able to see where the, let's say, cysteine, sulfoxide, and sugar sulfates are all located, and actually be able to identify small microsymbiotic organisms based on their sulfur speciation and those differences that form in these particular tubules. So it's a really powerful technique to be able to really visualize all the forms of, in this case, organic sulfur and how it's distributed within your sample as well. And now, if we want to bring this out to the more geological point of view too, we've also been looking at different pyrite samples from the Archean. So these are right at the rise of oxygen about 2.5 billion years ago. And by looking at this with the microprobe, we were actually able to find these really tiny micro inclusions within the pyrites. And these happen to then also correlate with other techniques that we used using nanosims, looking at isotopic ratios, and micromagnetics, so microsquid magnetics. And all three of these techniques combined together can show you where you have these really interesting mystery pyrites, so to speak, that have a odd magnetic field and an odd isotopic ratio. And this really then begins to lead you to see that there are other processes of transformation that have been occurring in these samples over time. And that may lead to us thinking that these pyrites aren't as pristine as we might want them to be for looking at proxies to go into the past. I want to finish up here by going through some of our other large programs. One of the things that we also have specialized at SSRL is a very large format beamline. So we have spot sizes here from about 10 microns up to 200 microns. But the stage here can be up to 1 meter in length. So we can do entire cores of samples. These are big mounts. And the idea here is to be able to look at microsites of activity, for biological activity. That might be an otherwise oxide core, looking for preserved traces of anaerobic activity. And be able to characterize this on the whole column scale, looking at meters of core, taking these for subsections, looking at smaller bits to really detail the micron scale type chemistries, and then even taking these into fib sections to take to our Beamline 6 transmission X-ray microscope to look at the nanoscale integration of these materials. And ideally be able to pull things from that really nanoscale up to the large core scale to make these types of interpretations on how the ecosystem may be developing these types of microsite capabilities. I also want to mention we've been working on a set of high energy resolution fluorescence detection, or commonly known as HERFT. It's a horrible acronym, but we go with it anyways. And this has been supporting a actinide program that we're working with at SSRL. And you can see that the HERFT spectrometry gives you that high resolution, allows you to really be able to probe the electronic structure much better in your near edge data. That allows you to really make high resolution maps to be able to look at differences in oxidation state that might be blurred otherwise, or very, very too similar to really separate out. And this is, again, developing for a large capabilities to be able to expand this. And we have a large library of other crystals that we use from our other beamlines. And this is now in user commissioning and will hopefully be in more broader use in the coming year. Peter mentioned some multimodal spectroscopies. And we like to do a lot of that with our beamlines at SSRL. So all of our beamlines, in terms of the imaging, in addition to that laser rangefinder, we also have optical microscopes. And those are all coupled to a UV-Vis spectrophotometer. And we can collect the UV visible spectrum at the same locations as our X-ray data, and actually be able to characterize color, beam damage, and other things that might occur. I'm almost done here, so I'll wrap this up quickly. We also have a FTIR microscope in the laboratory. So to kind of give us the full range of the photon spectrum, from X-ray to visible at the beamline to doing infrared offline, and try to correlate these on all of our samples together. And this got some of our first usage installed this year during our shutdowns. But we're doing XRF on samples that were COVID related and correlating to the FTIR maps at the same time. We've done a lot of things since we were in our shutdowns, but a lot of things have been really made for remote access. Multiplexers and easy to use data collection for people to do their experiments from their home or from their bedrooms even. And this is kind of a new, I think, a new paradigm of synchrotron research coming up, that a lot of us will be doing things highly remotely in the future. I'm just going to finish up here by saying that I hope you kind of see the excitement of what you can do by looking at spatial resolution as well as the chemistries. And just mention that there's lots of other opportunities for geoscience users. Many synchrotrons provide some sort of summer school for information about data collection and analysis. And I want to throw out a promo. There's typically a SES science synchrotrons in environmental science workshops that are done every couple of years. We didn't want to overlap too highly with this workshop. So the next SES 8 will be in fall 2022. And we'll have a series of geoscience workshops like this to introduce new users and such to these types of techniques, and have lots of hands-on work at the beamlines themselves too, or even virtually. And then I'll finish just by thanking all the lovely people at the beamlines. For more information, you can check out samsxrays.com. And we have a very nice, cool new 3D tour. If you want to take a look around our floor and see the different parts of our experiments, and thank all the people that have participated for data in this, and all of our sponsors. And I can take maybe half a question if there's still time left.
Well, there really isn't. We're at 1:06, and I'm trying to keep as strictly on schedule as possible, Sam. So I'm just going to thank you for a really wonderful overview. There is a question to you from James Green in the chat. Maybe you could just respond to James that way. That would be great. That was really interesting. Thanks so much.